I'm going to switch to this. And next, I want to introduce Dr. Lee, who flew all the way from Korea. And just kind of like to say that Dr. Lee's papers kind of like inspired my theoretical work in humans. So kind of like really thank you for that. And
Okay, thank you. Oh, yep. Yeah. Oh, okay. Oh, fish.
yes.
Yeah. Oh, do I need this microphone? I can see here.
Okay. Oh, you don't need the notes.
All right. So thank you for the organizers and chairs, and it's really great to be here. So yes, I flew all the way from South Korea to attend this meeting. I'm flying back on Monday morning. My lab is working on actually mouse models of RASopathies like NF1 and Noonan syndrome and CFC. And mostly we're focusing on the learning and memory phenotypes, but recently we are expanding our work to other domains like sensory processing and the motor coordinations too. But today I will mostly talking about the learning and memory stuff. So the disclosure, I have nothing to disclose, but except that I'm still suffering from jet lag. So, well, but I try to keep time. So, all right. So, oh yeah. All right. So we all know the RAS pathways, and this is very complicated pathways, right? So then, um, yes, the RAS pathways involve the multiple biological processes from like cell growth, differentiation, and many other things. And mutations in these pathways are associated with multiple disorders, not only cancer, but to the developmental disorders. That's why we are all here to study the RASopathies, right? So, and importantly, the reason I'm interested in these pathways, the RAS pathway is important for normal brain development and the normal brain functions like synaptic plasticity and the learning and memory and many other brain functions too. So actually, I put these very complicated pathways, but to show that this is not really happening in our real brain. So I always says that this kind of picture is kind of unicorn. Of course, these are all real, but I'm not quite sure if all these regulators are presenting exist in the same cell types. That'll be really the topic of my talk. So this is more simplified version of the canonical RAS pathways. And yes, I don't really need to explain all different diseases, but because whenever I give a talk to the different community, my lab has been mostly working on the Noonan syndrome, but even actually my previous mentor even pronounced it like Noonan syndrome. So I have to correct him every time. So, but here I don't need to do that. So, but because they share the same pathway and mutations in this pathway, mostly they increase the activity of the RAS-MAP kinase pathway. So we believe that they share the same pathway, so they share the many symptoms. So we assume that actually they're gonna share the many other like cellular phenotypes or cellular mechanisms too. But we'll see actually how is really what's really going on. Oops. So yes, they are highly associated with multiple cognitive symptoms, and also recently, the recent studies shows that these RASopathies are also highly associated with the autism spectrum disorders too. So maybe the NF1 might be the most well-known, the RASopathies, and in addition to these somatic symptoms, actually, the RASopathy, NF1, they also shows the multiple cognitive symptoms, although they don't really affect the global symptoms, but they have shows the phenotypes in like executive functions or like tensions and also the learning problems. And there's some patients also shows the seizures too. But I did my postdoc with Alcino Silva's lab, and Silva lab was using the NF1 heterozygous knockout mouse to study the mechanism underlying learning and memory deficit associated with NF1 for a long time. And they found that this mutant mouse shows that hippocampal learning and memory deficit. Also, they show these changes in the synaptic plasticity in the hippocampus. But maybe one of the most important findings from this NF1 heterozygous mouse was that we all know that in the brain, there's a excitatory synaptic transmission and the inhibitory synaptic transmission. But somehow, although NF1 is expressed everywhere, like including the neuron and glia, but somehow deleting NF1 in the mouse selectively affect the inhibitory synaptic transmission without really affecting the excitatory synaptic transmission in both cortex and the hippocampus. That was kind of one of the major findings in like 10, 15 years ago. So then I started working on the Noonan syndrome mouse models when I was in Alcino's lab. So yes, Noonan syndrome is actually more common than the NF1. That's actually most common genetic disorders. And then these associated with the mutations in these genes, they are all associated with RAS/MAP kinase pathways. And then they have these clinical symptoms, including the heart problems and short stature and growth delay. And then they also, some of Noonan syndrome patients, they shows these learning disabilities and also autism-like phenotypes. And in the mouse models, we found the hippocampal learning and memory deficit when we tested this mouse in the Morris water maze, test the mouse swimming in this in the murky water to find the hidden platform. So this wild type mouse already find the platform, but the mutant actually takes longer time to find and the hidden platform here. So we used the PTPN11D61G mutant mouse that generated in the Benjamin Nils lab. And also we tested actually N308D mouse too, but the N308D mouse shows the milder phenotype. And just to summarize this finding, we found that this spatial learning and memory deficit and also changes in the synaptic plasticity. But importantly, in contrast to the NF1, which shows the changes in the inhibitory synaptic transmission, this Noonan syndrome mouse model shows the selective changes in the excitatory synaptic transmission. So, and then expressing this mutant only in the excitatory neurons can recapitulate the multiple phenotypes shown in the whole body knock-in mouse models. So that really leads us to the central question in my lab, because NF1 and PTPN11, as I showed you, they are in the same pathway. These mutations, they all unregulate RAS/MAP kinase pathways, and behavior symptoms, in terms of the cognition, the behavior symptoms were similar, but why the NF1 affect the inhibitory synaptic transmission, but the PTPN11 mutation affect the excitatory synaptic transmission? That was one of the central questions in my lab. So our hypothesis is that although we draw this RAS/MAP kinase pathway, everything squeeze into the one page, maybe in the reality, maybe the signaling pathway might be distinct among different cell types, like the green blue cells excitatory neuron and red cell is GABAergic inhibitory neurons. Maybe their signaling network might be quite different. So to test this idea, this was like almost 15 years ago. That time we didn't have single cell RNA-seq technology. So we generated this reporter mouse expressing this fluorescence protein in the excitatory or inhibitory neuron in mouse brain. And actually my student manually picked up all these cells, and then we collected this cell and performed the RNA-seq to see to analyze the RAS/MAP kinase pathways in different cell types. And to make a long story short, this is our finding. So these are all the main regulators for RAS/MAP kinase signaling pathways. And these genes are enriched in the excitatory neurons, but these genes are enriched in the inhibitory neurons. So we were able to reconstitute the RAS/MAP kinase signaling pathways in excitatory or inhibitory neurons in mouse hippocampus. And then here, for example, we found that and F1 seems to be enriched in the inhibitory neuron in mouse brains. So that may explain why the NF1 deletion have bigger impact on the inhibitory synaptic transmission rather than compared to the excitatory synaptic transmission. This might be just too simple-minded explanation, but this was what we are finding. So like SYNGAP was enriched in the excitatory neuron, etc. So, and we also got the human cortex tissue from our hospital, and then we found that NF1 is also enriched in the inhibitory neuron compared to the excitatory neuron in the human brain too. But then what about the PTPN11? Because we found that selective changes in the excitatory synaptic transmission in the PTPN11 mutant mouse. So initially we assumed that maybe PTPN11 is enriched in the excitatory neuron, but that was not. Actually, PTPN11 shows that both the neurons shows compatible expression level of PTPN11. So initially there was a big disappointment, but when we look into this signaling pathway more carefully, we knew that from the other studies, we knew that the PTPN11 requires other adapter proteins like GAP1 or GRB2 to be coupled to the RAS signaling in the other cell types. And we found that these proteins are enriched in the excitatory neuron, and then their expression level was quite low in the inhibitory neuron. That's why, although we have a PTPN11 mutation in both cell types, they cannot really activate RAS signaling pathways in inhibitory neurons. So later we were able to actually block block this interaction, then we were able to rescue these phenotypes and cellular phenotypes, and also we were able to rescue the behavioral phenotypes in mouse models. And also in another experiment, actually, we were able to overexpress this GRB2 and GAP1 in the inhibitory neuron. Then we can switch this inhibitory neuron into like excitatory neuron-like state. So that was actually a quick summary of the first part of the story about this Noonan syndrome mouse models. We found that this RAS signaling pathways are quite distinct between the excitatory and the inhibitory neurons. And this study was mostly done by this Hyun-hee and Taehyun in my lab. And then later, but we know that actually in the brain, we have more glias than the neurons. And here, this is the immunostaining of mouse brain. And all these yellow cells are labeled astrocyte, which is kind of the glia cells. Before, we believed that when I was a student, just textbook says the glia is a kind of glue, just filling the gap, or they play the supportive role in our brain. But now, actually, we know much more. Actually, the astrocyte plays, or in other glia like microglia, they play more active roles controlling in brain functions. And another student in my lab now, she's a postdoc. She actually wanted to study the contribution of this astrocyte in the RASopathies. So again, we switched to study the CFC syndrome models. One of the reasons is that the Noonan compared to the Noonan syndrome, the BRAF mutation associated Noonan or the CFC syndrome are associated with more severe cognitive symptoms. So, and then almost in most of them show the cognitive phenotypes and intellectual disabilities, and they are associated with the MEC1, of course, but also associated with the BRAF mutations. And again, in collaboration with the Benjamin Neal's lab, we generated this conditional knock-in mouse that we can express this gain-of-function mutation, K499E, in the selective cell types. And of course, first, our favorite cell type was excitatory neuron. So we expressed this mutant BRAF in the excitatory neuron. But surprisingly, actually, this is the water maze test again, but we did not see any phenotypes, although we see the huge increase in the MAP kinase activity in the brain. So still, this is kind of mystery. So, but we tested actually a lot of this mouse in many different behavior tests, and also we tested the synaptic plasticity, and then we did not see the phenotypes. And my there was a big disappointment. But then I convinced my student maybe just this BRAF is more like NF1. Maybe you should look into the inhibitory neuron. So my student crossed this conditional mouse to the inhibitory crine lines to express this in the inhibitory neurons. And again, then we did not really see any significant deficit in the hippocampal learning and memory, and also social behaviors and other behavior tests too. So it's been already like, I don't know, more than 3 years. So she almost quit the lab. But then I convinced her again, okay, we should express this. Yeah, also other cell types too. Then we cross this to the nesting cree that to express this almost every cell types in the brain. And finally, we see this huge learning and memory deficit. So this mouse, this is the biggest phenotypes that I ever seen in my whole career. So then we cut the brain to see what's going on. This is another behavior test. And again, we see that the memory deficit. And when we cut the brain, then we were surprised to see there's huge changes in the population of the astrocyte. These green cells are astrocyte. And normally we don't really see the activate the astrocyte in the wild type, but we see this activate the astrocyte everywhere. So that was a big surprise. And then we did RNA-seq and found all these genes are all related with the reactive astrocyte. They are changed. And then we, in collaboration with the other group in Korea, we made a knock-in, the human organoid expressing the exactly same mutation. And then again, we see the huge changes in the astrocyte and also similar changes in the gene expression. And the ARC activity went up in the astrocyte. And then later we have in this hospital, we have the 4.5 years old boy harboring the exactly same mutation. He has a heart anomaly, and then he also had the feeding problems, and he shows the severe developmental delays, and then he has this same K499E mutations. And then we generated the iPSC from his blood, and then we grow this into the organoid, and we found exactly same finding. We see the huge increase in the GFAP positive astrocyte. So I think this astrocyte phenotype is really conserved from the mouse to the human. And then to find to test this is really sufficient to this mutation in the astrocytes is sufficient to induce the phenotype. We express this mutation selectively in the astrocyte in the adult mouse, and we were able to mimic the phenotypes that the mouse shows the severe phenotypes, and also we will recapitulate the cellular phenotypes too. And finally, just to see, oops, so we can skip this. So then we also find that actually calcium activities increase in the astrocyte. And then, yeah, we all know that MAP kinase is important. So we suppress the ARC activity selectively in the astrocyte. And then we were able to reverse the cellular phenotypes. And also we were able to reverse these behavioral phenotypes. And so this means actually this signaling in the adult astrocyte is really critical for these behavioral phenotypes. And then finally, since we found that astrocyte calcium level is increased in this mutant mouse, again, another manipulation, we reduced the calcium concentration by expressing these calcium pumps, again, selectively in the adult astrocyte, then again, we were able to rescue the phenotype. This is the mutant mouse. But then when we normalize the calcium level, they selectively search for the platform. So again, with this is the summary of the second part. We show that this gain-of-function BRAF mutation really increased the reactive astrocyte in the brain. And then we were able to actually normalize this behavior phenotypes by normalizing MAP kinase activity or calcium activity in the astrocyte. So with this, I like to just take a message is that, yes, we were talking about this using the MAP kinase inhibitor to as a treatment. But I think when we consider the treatment, we really need to consider the cell type specificity, because these different mutations affect the different cell types. We don't really want to affect all the cell types. I don't know how to do that. I have no idea, but I think we needed to find a way to be more specific. With this, I'd like to just thank all my lab and collaborators, and then, okay, thank you. I'll be happy to take any questions.
Bruce.
Gorgeous work. We're all glad your graduate student didn't kill you. So in a way, this begs the question still, where does the specificity come from? Because you've now given us 3 examples. They've hit 3 different cell types, and yet they're all gain of function in the pathway. What are your insights into the why here?
Right, that's really, really important question. So when we were working on NF1 and Noonan, so we thought we understand, we knew the answer, because when we look into this different network, we were able to find out this differentially expressed component. But actually for the astrocyte, we don't know. Actually, that was still mystery, because that was still BRAF was fully functional in the neurons, but I don't know. We didn't have a phenotype, so we don't know actually.
Pao?
Ari, maybe this is piggybacking on Bruce's question, but have you now considered maybe doing similar analysis to your RNA, but maybe at the protein level? Because maybe that's where some of the differences
I
lie
see.
in-
That's also a good some So we are actually doing some proteomics work too, also phosphomics too. So maybe we get to know something.
A very beautiful talk. So I'm kind of interested whether you have tested using anti-inflammatory drugs to see whether you can reverse the phenotype.
That's also a good comment, actually. No, we plan to do it, but we haven't tested it yet. But we didn't see any changes in the microglia. So
That's fascinating. One more question? Yes.
Great work, Yong-Seok, as usual. One question I have, which you may or may not have the answer to, is so within your RNA-seq data set, have you looked at different isoform usage for the different cell types? And whether sort of potentially part of the explanation may be simply, within specific hotspots, you may see specific isoforms that are more prevalent in one cell type versus the other, and that may actually explain your phenotype.
That's also a good comment, because we just finished also single cell RNA-seq with this BRAF mutant, so we can actually take a look at it. Thank you so much.
I'm seeing a neural lunch forming here. So, okay. Thank you so much.
Thank you.